Hello everyone, welcome to my channel by Universe. I am Suyanka and today we will learn protein targeting, also known as protein sorting. Protein targeting is the biological mechanism by which proteins are transported to different cell organelles from cytoplasm. As we all know that protein synthesis occurs within the cytosol by ribosomes. These proteins must be translocated to different cell organelles for their specific functions. Translocation of proteins occurs during translation known as co-translational translocation, and translocation occurs after translation is complete known as post-translational translocation. Protein transport across membrane of different organelles occurred in different ways by different organelles. In today's lecture, we will learn about how the protein are transported into nucleus. Before that, please subscribe my channel. Nuclear envelope in all eukaryotic cells have nuclear pores that form the gates through which proteins enter or exit the nucleus. A nuclear pore is a large, elaborate structure composed of complex of about 30 different proteins. This complex is known as nuclear pore complex. The proteins that make up the nuclear pore complex are known as nucleoporins. The nuclear pores complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. Nuclear pore complex consists of an assembly of 8 spokes attached to the rings on the cytoplasmic and nuclear sides of the nuclear envelope. The spoke ring assembly surrounds the central channels containing the central transporter. Cytoplasmic filaments extend from the cytoplasmic ring and form nuclear basket. Import of nuclear protein. Nuclear protein in cytoplasm has lysine-rich amino acid sequences which is known as nuclear localization signal. Nuclear localization signal helps to direct nuclear proteins to the nucleus from cytoplasm. Importin. It is a cytosolic protein and receptor which binds with the nuclear localization signal of nuclear protein in the cytoplasm and release it in the nucleus. RA and protein are small monomeric nuclear GTPs which are present in nucleus. When importin and nuclear protein complex entered nucleus, RA and GTPase binds with importin and nuclear protein complex and cause conformational change in importin. This conformational change causes importin to detach from nuclear protein. Importin is now attached with RA and GTP. RA and GTP helps importin to exit from the nucleus. The cytoplasmic side of nuclear pore complex has RA and GAP which causes the GTP to be hydrolyzed to GDP. Importin dissociate with the RA and GDP and ready to attach with different nuclear protein from cytosol to nucleus. NTF2 are the cytosolic protein which are responsible for the nuclear import of RA and protein. RA and GDP attach with NTF2 which helps RA and GDP to enter nucleus again. The nucleus contains RA and GEF which convert RA and GDP to RA and GTP by hydrolysis process. RA and GTP is now ready to bind with next importin complex entered in the nucleus. In this way, import of nuclear protein occurs. Now let us see how export of nuclear protein occurs. Some of the proteins which synthesized in the nucleus has to exit from the nucleus to cytoplasm for the specific function. These nuclear proteins has leucine-rich amino acid sequence known as nuclear export signal. Exportin are nuclear protein or receptor which binds with nuclear export signal of nuclear protein with the help of RE and GTP to exit the nuclear protein from the nucleus. Exportin nuclear protein and RA and GTP complex brings nuclear protein out of the nucleus in cytoplasm. RA and GAP present in the nuclear membrane convert RA and GTP into RA and GTP by hydrolysis, which dissociates exportin from the nuclear protein, and nuclear protein are free in the cytoplasm. Exportin and RA and GDP complex return to the nucleus by the help of anti-F2. RA and GF present in nucleus converts RA and GDP into RA and GTP by hydrolysis, which cause exportin to dissociate from the RA and protein. Exportin is now ready to attach with new nuclear protein in nucleus and transport it into cytoplasm. In this way, protein targeting into nucleus occurs.